Hi, my name is Maria Hudak, and I'm an aspiring biomedical engineer. Let me tell you about something that really excites me: tissue engineering and genetic engineering. Specifically, you need to hear about hydrogels and Cas9 CRISPR complexes. Let me start with hydrogels. Hydrogels are interwoven mats of tiny fibers that make up a sponge that can absorb a large amount of water. It can be either like your kitchen sink sponge or a sponge cake. The kitchen sink sponge would be if the molecules were joined with covalent bonds, which means that no matter how much water it absorbs, it's probably not going anywhere. Meanwhile, you can have organic molecules that are only bonded with secondary interactions like van der Waals forces and hydrogen bonds. These might eventually break apart, like a sponge cake that's absorbed too much water. Now, the applications of these are incredible. Suppose that you could add tiny little holders along every single strand of your sponge. These would be receptors. They can hold any kind of molecule. You can attach them to your gel, load the different molecule that you'd like delivered somewhere, place it anywhere in the body that you need that molecule delivered. A trigger hormone or release chemical releases those drugs exactly where you need them. This is a much more reliable method than injecting or ingesting that drug and hoping that the circulatory system will get it where it needs to be. Another application would be to stuff your sponge full of different cells and make it sort of like an artificial organ. You could circulate body fluids through it, kind of like a dialysis machine. What if you could filter leukemia from the blood just like a pool filters bugs from a swimming pool? Now, let's switch gears and talk about Cas9 CRISPR complexes. You've probably already heard of the different types of enzymes that can cut DNA, but they only respond to very specific base pairings. Cas9 is a type of enzyme that's non-specific. It doesn't have a pre-programmed binding site, it just is functionalized once it combines with an RNA strand. And since humans can manufacture any RNA strand they want, that means you can get it to cut anywhere you want. Cas9 has a special affinity for wherever it bonds. That means it cuts closer to stoichiometric equivalence, and that means you're going to have smaller amounts of cutting in much more specific places. When you're cutting DNA, that means you can cut out bad genes and insert good ones. Now, the really exciting part is when you combine hydrogels and Cas9. Hydrogels can deliver small molecules anywhere we want in the body. Meanwhile, we have Cas9 that so far is really only valuable for injecting on a single cell basis. But what if we could deliver Cas9 to any specific cell at sites in the body with a hydrogel? Or what if we could filter cells in the body towards Cas9 stored in a hydrogel? Can you imagine the incredible potential of being able to edit genes on a much larger scale and in a somatic cell? That's why I'm really excited about these two technologies. I can't wait to see where they go in the future.